60 to 80% of the genes in worms are homologous to us humans. And we don't like to think about that, but that is evolution, I think, at its best.
Welcome, everybody. This is Buck Joffrey with the Longevity Roadmap. Interesting conversation today. Dr. Heidi Tissenbaum. She's one of the sort of cutting-edge researchers, with C. elegans, which is a worm that is often worked on in the context of longevity research. She's one of the pioneers in that space. A really interesting conversation. We talk a fair amount about, listen, this is for science geeks, right? Like to help you understand for example, why we use worms in the first place, what they tell us, some of what those key initial studies showed us regarding IGF-1, which you may know seems to be a major player when it comes to longevity across species. The role of stress, both cellular stress, and sort of the general stress that we kind of deal with on a daily basis, trying to differentiate between those. We talk about hormesis, and ultimately I try to get a perspective on Dr. Tissenbaum's view on sort of the future of longevity and longevity science. And what I think is interesting is that what you'll find is that she's not as optimistic as others, but I would like to point out that we've had plenty of people in the same space, literally in the same lab. She's in the same lab. She did the same postdoc mates included, Matt Caberlin and David Sinclair. And she, she's come out with different opinions on that. She's not as optimistic as those guys who, Sinclair in particular talks about how, age reversal is already possible. Whereas, you'll hear Dr. Tissenbaum say, something to the effect that she doesn't think we're there at all yet, although she has left the door open for various types of interventions and research that could at some point lead to those things. At any rate, if you like science, if you like hearing about the role of some of these key pathways such as IGF-1 in the aging process, you're going to enjoy this interview, and we'll have that right after these messages.
Hey longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com.
Welcome back to the show, everyone. Today, my guest on Longevity Roadmap is Dr. Heidi Tissenbaum. She's professor of molecular cell and cancer biology at UMass Chan Medical School. She completed her graduate work at Harvard Medical School with Dr. Gary Ruvkun, and her postdoctoral training with Dr. Leonard Guarente at MIT, who you may recognize these days as kind of a big name around the supplement industry, and also where David Sinclair did his PhD as well. So that must have been an interesting time. Dr. Tissenbaum is internationally recognized for her work using C. elegans to uncover the genetic and molecular pathways that influence lifespan, particularly those involving insulin and IGF-1 signaling, diet, and stress. Her research is helping connect the dots between the fundamentals of biology and strategies to promote resilience, metabolic health, and healthy aging. Welcome, Dr. Tissenbaum.
Nice to meet you. I'm happy to be here.
So why don't we just start out with this? I mean, for our audience who they probably hear about worms and C. elegans, often when you hear about basic science research, especially when it comes to longevity, why choose worms and C. elegans in particular as a powerful way to look at this kind of biology?
That's a great question to begin with. And it seems kind of strange when I tell people I work on worms, they kind of close their eyes. But if I tell them I work on longevity, they are very, very interested. So I've had, 30 years of answering this question, and the real reason is the worm, C. elegans. Everything is done under the microscope, but the whole worm is 954 cells. And what was the Nobel Prize was awarded to several, probably 20 years ago, to Bob Horvitz, John Sulston, and Sidney Brennan for discovering the worm. And what they did is they did meticulous uh, lineage tracing. So they know from one cell what happens to that cell as it divides to 2 to 4, all the way to 954. And now we can ask questions how one cell is different from another, which you could never do in a complex system like a human or even a mouse. But with 954 cells, you can actually ask what makes one cell different from another. And so that is the basic uses of the worm. But for longevity, the worm lives about 3 weeks. And when you're a graduate student or a postdoc or a fellow or even a researcher, that makes a huge difference because you can do many, many experiments in a short amount of time, which you cannot do in a more complex system. Moreover, the average lifespan is very tightly regulated, so you can find modifiers of lifespan as little as 10 to 15% different from the average. And that is not, that makes it very easy to identify modifiers of lifespan. The other thing that is kind of hard to believe, but depending on what you read, 60 to 80% of the genes in worms are homologous to us humans. And that is, we don't like to think about that, but that is evolution, I think, at its best.
Yeah, that's great. Well, let's, in terms of this model with C. elegans, what kinds of things have we've been able to reveal about the fundamentals of aging that we might have never discovered if we were just using mice or tried something silly like using humans for that?
Okay. So that's a really good question as well. And I think that gets into the insulin IGF-1 signaling pathway. So when I was a graduate student, we discovered that several of the genes we were working on were, when we identified them molecularly, and this was in the laboratory of Gary Ruvkand, who recently won the Nobel Prize, I might say, but not for this work. um, Anyways, so what we discovered was that genes that we were working on in the worm, and Cynthia Kenyon had previously identified one that was associated with lifespan, as well as Tom Johnson had done the original work on the gene that age one appropriately named. And we discovered when we cloned them, they were part of an insulin IGF-1 signaling pathway. And within a decade, about 8 to 10 groups had identified one of the genes to be associated with longevity in humans. So I think that's sort of what brings worm aging to light, in the fact that what we were studying in a little crater that only lives 3 weeks goes to identify something associated with human longevity across multiple populations within just a decade. So that's the first thing. The second thing is the whole field of sirtuins, which is sirtuin biology, which I think some of your audience might be knows about. That began with some of my work when I was a postdoc at MIT to show that basically extending of lifespan of SIRT1 also occurred in worms, not just in yeast, as our laboratory had previously shown. So those are two major finds that are relevant to longevity, to the longevity field.
Yeah. Who was that? Do you ever post that? Was that Lenny Garante's
Yes,
lab?
yes,
Okay, interesting, interesting. So he's
yes.
gone on obviously in a big way in this longevity space as well. So let's drill down on IGF-1. I mean, it's pretty important, right? So, can you walk us through what you found with worms that might be sort of a universal thing that that pathways turned up or turned down in worms?
So originally we discovered the genes that we were working on were also important for a process called diapause, which is sort of like a spore or sustained in long, harsh environments. And these genes also worked on that. And what we found was that we basically identified all members of a signaling cascade that begin with a receptor that is homologous to both insulin receptor and IGF-1 receptor. So in mammals, you have both insulin receptor and IGF-1 receptor. In worms, it's just one receptor that signals down a canonical signaling cascade into ultimately into a fork and transcription factor, if you want to be detailed, but into something that binds DNA, and it modulates the whole pathway modulates the gene at the end going in and out of the nucleus, so it could turn on or turn off genes that are required for whatever, for metabolism, longevity is associated with that, but all these other genes.
Yeah, yeah. So basically, the idea is if you can down-regulate IGF-1, it basically, it creates a cascade of events that seem to extend life. Is that right?
Exactly.
Right.
Exactly.
Yeah.
Exactly.
So, you also studied stress resistance. So, why is the ability to handle stress such an central determinant of longevity?
Well, in my opinion, from all my reading, I think that what stress does is if you lower your stress, you're ultimately modifying an inflammation signal. So ultimately, the ability to deal with stress and to deal with it in a way similar to evening our levels keeps inflammation at bay, and you don't get a spike in any inflammation. Moreover, stress is linked to metabolism, and what we don't want is any strong changes in our metabolism. That's why, if I could just take a leap, uh, if we think about our eating and our diet, people want us to have 6 meals a day because then in my belief, I believe it's keeping our insulin levels constant. And if we don't, if we're getting back to insulin, if we don't keep these levels constant, it's not good for our health. And in worms, the reason stress is linked to longevity is because it's controlled through insulin. And we want to keep those levels constant, not have these big spikes in our levels because it's not good for our overall system.
Now, you mentioned something about 6 meals a day. I mean, that's sort of that's different from our, a lot of the longevity people out there talking about intermittent fasting, various fasting. So how do you, I mean, there's plenty of evidence that there is, at least in the short term, benefits to people in terms of metabolism, in terms of metabolic syndrome and that kind of thing. So how do you put those together? Because I guess the other question is, when you talk about stress, particularly cellular stress, there's also the concept of hormesis, right? So some level of stress, which we know is induced by fasting, can be beneficial to us. And some people think that may actually trigger some of the longevity genes. So I'm curious on your take on all this.
Okay, so there's a lot to answer.
Yeah,
So
sure.
first, hormesis. So hormesis is as you know, is the fact that if you give a little bit of stress during development or even in early adulthood, and then you re-stress the animal the same way, it survives much longer. And it's quite easy for us to do a variety of stresses in the laboratory and see that occurring. Okay, but so to me, what all of that does is it's like a vaccine, like when you get a little bit of, when you're getting vaccination, that you're able to survive stress better. But it's all, I think, what that does is it modulates your ability later. You're able, and you don't get a sudden spike in your stress response. So that's what I think it is. It's all about modulation. Now, intermittent fasting, it definitely works. It's something many people do, and I believe it can work for a lot of people. Um, but again, I think what intermittent fasting does is it decreases the total amount of food that you eat. So you don't get a huge bolus of insulin at one time. And I think that's what's really bad is it's the constant shocking of your body. Like I even think of I live in a cold climate, so I even think about when we're cold outside and then we go into an overheated building in the winter. That's not good for your system. I just think constant stress is bad for your overall metabolism and your ability to survive long term and deal with stress.
Yeah. Yeah. Can you, just for the audience, kind of just make the distinction between cellular stress and the concept of, again, hormesis being like, what the simplest way to say it is what doesn't kill you makes you stronger, right? I mean, that's essentially it. And so,
Tell my students.
yeah, so a lot of the things that we hear about in this space, you mentioned cold. I mean, I don't know that there's any evidence that these ice baths or anything like actually do anything for longevity. They may help with dopamine release and that kind of thing, but certainly saunas, right? Sauna, there's a tremendous amount of at least data from Finland and all that. Again, the idea being potentially that we're sort of shocking the system a little bit. We're stressing it. You actually brought up sort of the change in temperature. I would think that from what the sauna data shows us, that kind of changes in temperature actually might be good for us. No?
I'm actually unaware of the sauna evidence.
No. Oh, okay. Yeah. Yeah.
I just have to say that in longevity research, whether there is so much data out there because everybody, especially as we age, everyone thinks about this more and more and more. And I'm sort of at the other end of the spectrum where I want evidence and I always go to read the primary articles. And it's very hard to keep up in the field.
Oh, 100%.
It's very, very hard. And so
no, and I totally get that. And if it were anecdotal, I wouldn't bring it up. The Finnish data is pretty robust. It might be something I would be curious to hear about from you at some point if you had a chance to look at it. But
I would be happy to look it up. But I can comment on cellular stress versus organismal stress if that's what you were interested in. So, just for the audience, when people talk about cellular stress, they're really talking about what happens in our cells directly. So, people will notoriously do experiments in cell culture. So, they'll have cells on a plastic petri dish growing. And sometimes these cells are primary cells from human tissue, and sometimes there's cells like you might have heard of HeLa cells, Henrietta Lack cells that are transformed. And so they will go on and on and on and on in culture. No, I don't study that. What I study is organismal aging, let's say, not cell aging. And so when I do lifespans or all the experiments that I've ever done are just like aging, just like you and me. How many days does the animal actually live? Okay, that's organismal aging. And there still is some differences between people that study cellular aging or cellular senescence and organismal aging. So, but something that I think that your audience is interested is what is the difference between lifespan, which I just said that, and healthy aging or health span. And we could talk about that as well.
Yeah, sure. Well, let's talk about that.
Okay. So about 10 years ago, and I'm talking about myself. I'm not tooting my own horn
Yeah, no,
here.
sure.
Okay. So probably about 15 years ago, I went to a conference, and about 10 years ago, we published a paper where we were looking at health span in long-lived worms. Now, health span, or people acquainted as healthy aging, but in a laboratory setting, I look at it similar to lifespan. So it's a span. How healthy is an animal over the course of its life? Because the good and the bad is lifespan is a defined measurement. Anyone could go to any laboratory that's studying organismal aging and ask, what is the lifespan of that animal? And everybody knows what that means. Okay? It's simply how long the animal lives. So for about 30 years, many people successfully did really good work showing chemicals, genes, regimens that extend lifespan. But the question always became, what is the quality of that long-lived animal? Are they increasing a frail segment of their life, or are they actually healthier for a longer period? Because if you think about it, if we're leaving healthier till 80, but now we have 40 years in a nursing home, that's not good for society, right? I mean, it's unsustainable. So many people started speaking about healthy aging or health span. The problem is there is no universal definition of health span. And if you type, anyone in your audience types healthy aging into the Google browser, you will get pictures of gray-haired people smiling. That is what, I'm sorry, but that's what you get. And so how do you define healthy aging or health span in the laboratory first, in an animal second, and in a worm third?
Right. So you're saying there's no gray-haired worms out there, doctor? Are you?
No. No, and then moreover, I mean, people do things like look at gray-haired mice and see a reversal of that. But, I don't know if you just aged a mouse normally, what would it look like? Like, exactly what you're saying about a gray-haired worm. Like, what normally happens? Not every human gets gray hair at the same time. Not every human loses their ability to be mobile at the same time. Not every, it's individual. We could assess it overall as a population, but it's individual. So the good news is that in the longevity field, many people have started to put in measures of health span in their manuscripts. And what people typically look at is mobility and survival and ability to survive stress. And those are two things I think that are really good to look at. We've looked at it extensively. So I believe that those are good. The ability to bounce back from a stress as you age, the ability to move. Either we do both forced movement and unforced movement. Those are, I think, very good. I think for people, they also measured their ability to walk, ability to stand up, so you're not frail, basically.
Right. In humans, whenever you talk about IGF-1 in particular, you're invariably enter into conversations that involve growth hormone. And also, one of the ways that you can you ultimately increase your IGF-1 most potently is actually by weightlifting. How do you bring these things together? Because it's, people think about, well, in humans, well, maybe I should be trying actively to decrease my IGF-1. And well, growth hormone's no good. But the problem is that every time I go and lift weights, I am increasing my IGF-1. And in that case, but I'm supposed to be lifting weights because there's an enormous amount of literature that says that is good for me. So how do you put all these things together for like somebody listening?
I think it's very difficult. I think that, my motto is, from my reading, and of course I don't study humans, but from my reading, weightlifting is the single most important exercise as you age, because you get frail, and it kind of goes against a lot of signaling pathways, but I believe that weightlifting is significantly important. And especially now with people like on all these Ozempic like drugs where you lose muscle mass, I think it's going to become more important.
Yeah. Yeah. Well, and that's in itself a big question. There's a trade-off of somebody who is 50 pounds overweight with poor glucose control and versus getting that under control and losing weight and then losing muscle mass. So it's
Right.
all a big trade-off, right?
Yes, yes. I mean, my motto really for human longevity is really keep your mind active and your keep your mind active and your body moving, and also avoid sugar. That's my
Yeah. No, I think that's a very good place. That's probably about 80% of it right there, right? So, um, other stuff, there's a lot of buzz these days about various drugs, supplements that claim to, mimic caloric restriction and therefore, modulate IGF-1. Do you look at anything that's out there, any drugs, any supplements, and have any sort of feedback?
So the problem with most of the supplements is they're not tested in the laboratory. There's no regulation of supplements. I mean, I personally, and then second of all, personally, I get skeptical when people's personal finances interfere with laboratory findings. So I'm very skeptical about a lot of the things out there. As I said, I think that, what I said in terms of keeping your mind and body active. And so for mental stimulation, even if, let's say, you retire whenever, 65 or whatever, and you're used to doing crosswords, don't just do a crossword. Throw in a Sudoku, throw in another card game, or throw in something else to keep those neurons firing and making new connections. That's intrinsically important. Then the other thing is there is something to genetics. Some families can, live into their 90s or even after 100 They usually have a brother or a sister that's also long-lived. And those people, for some reason, seem to be resistant to things like diet and smoking, all the things that we think are bad for us. For the majority of us, they are bad. But there are a few in the population who have genetics in their favor that they're long lived within their family. And that's something we don't understand yet.
Yeah. From your perspective as a basic science researcher on longevity, what in the big picture, I guess my question is, how far away are we from potential discoveries that will actually, impact health span and health span certainly is something that we're addressing already, but this idea that most people are still going to die before they're 100 years old. There are people in your field who think that that really is something that we can fundamentally change, that we can make discoveries that make us live, a material number of years longer in good health. What's your perspective on that?
I'm going to get into trouble. So I am skeptical.
You're skeptical, yeah.
Skeptical that we can do that. I mean, I don't know. In some ways, I feel like these GLP-1, the Ozempic-like drugs are longevity drugs because people would have died of heart disease or diabetes before. And now when they lose 50 pounds, all of a sudden their A1C is normal and they're no longer needed to go to the cardiologist or the diabetes clinic at the hospital. So in some respects, something like that could be a longevity drug. But I still, aging involves our whole body. So I think we're not at the point where we understand the fundamental key signal that regulates all our signaling pathways, all of our body. Like if we have something that works on our muscles, will our brain still be active? If we have something that works on our brain, will our body still be active? I mean, most of us know elderly people whose minds are fine, but their bodies aren't fine. And then, so I wonder if we can really do that on such a complex problem. And of course, that is the ultimate goal, but I don't believe we're understanding it yet. I believe we will, but not yet.
But you do believe we will, which is, I guess, kind of the fundamental question. And when you think about and I'm just asking you, kind of think in a big picture way, where do you think the most promising basic science research is about fundamental reprogramming? And there's tons of people talking about various things, whether it's, I just talked to somebody who was talking about who was an expert in both mutations in the genome and epigenetic drift and that kind of thing. Sort of similar take as you in that, like, that alone is not the golden, or isn't the silver bullet, but that there is ultimately something in the genome that, if they're able to figure out how to slow down mutations, DNA methylation and epigenetic drift, that could do things. So I'm just giving you that one example. It's a long question I'm asking you, but when you look at all the different things that are out there, like, what gets you excited? What makes you think, gosh, that could do it, or that might do it someday?
That's a really difficult question.
I'm sorry. That's what I'm here for.
Right. That's right. I'm really not sure. I think it's going to be a combination of things. But ultimately, it still comes down to the question of, how do you prove something like that? What are we actually looking for in a drug? Are we looking for something in a cell-based assay that's going to make the cells survive stress, and that's going to have a human intervention. How are we going to do something? And anything right now, if we have a problem, it takes 80 years or so for us to figure things out. Like, what actually are we looking for, and how are we actually going to prove that? That's where I come from. That's my pessimism is if we had that answer, how is that going to look in the laboratory, in the clinic, anywhere before we know that that's real?
Yeah, I get it. Fantastic. Well, I do appreciate your time today, Dr. Tissenbaum. It's been really great learning from you. And and again, thanks so much for being on the show.
Thank you very much. My pleasure.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.